Chapter 8. Pathogenicity. Pathogenicity is the ability to cause disease. Virulence is the extent of pathogenicity. Microbe versus host. To cause disease, a microbe must gain access to the host, evade host defenses, and damage host tissues. Steps to disease. First, it must gain entry through the portal or host. Adherence, invasion, so it must protect itself from the host defense system while invading the tissues, so while penetrating. And damage. Porous entry for pathogens. Mucous membranes. The respiratory tract is the easiest portal to enter and the most commonly infected. Many microbes travel in the aerosols which we breathe. Example, cold, TB, influenza, smallpox, and pneumonia. Gastrointestinal tract or the GI tract. Through food, water, and dirty fingers. It must overcome the low pH of the gut. Example, amoebic dysentery, hepatitis, shigellosis. Genitourinary tract for STDs. They could come in through the broken route, also known as the peritoneal route, or unbroken membranes. It depends on the microbe. Example, HIV, genital warts, herpes, and syphilis. The skin is impenetrable for the most part. This is not true if the skin is broken. Hookworm larvae can bore through your skin. Hair follicles and sweat gland ducts, which we know many of which have antimicrobial oils associated with them. For example, some fungi, such as ringworm, can infect actual skin. The peritoneal route is when there's a break in the skin and the skin can be penetrated. Every organism has a preferred portal of entry, so many microorganisms have to enter in a specific way and in a certain place to cause disease. For example, Vibrio cholera infects the GI tract. Therefore, rubbing an infected oyster on a wound will not cause cholera. Number of invading microbes. The ID50 is the number of microbes in a dose that causes infection in 50% of the organisms infected. So ID50 is the infectious dose for 50% of the test population. The LD50 is the number of microbes in a dose that kills 50% of the organism infected in a sample. The LD50 is the lethal dose of a toxin for 50% of the test population. So we're looking at how much we could kill, okay? So if we had ID50, if we had 100 people, we're looking to see if the actual dose that will infect 50 people. Same thing for the LD50. We're looking to see if we had 100 people, then we're looking to see how much people would be killed by a dosage of 50 people. The LD50 and the ID50 change for different circumstances. Let's take a look at an example. Bacillus anthracis has 3 portal of entries: the skin, inhalation, ingestion. The ID50 for the skin is 10 to 50 endospores. For inhalation, it's 10,000 to 20 endospores. For ingestion, it's 250,000 to 1,000,000 endospores. Which of these is most infectious? If you said skin, you are absolutely correct. It only takes 10 to 50 endospores to infect 50% of the population. So basically, ingestion, you have to ingest a lot more. Potency of toxins, in terms of LD50, the portal of entry would be botulism toxin is 0.03 nanograms per kilogram, sugar toxin 250 nanograms per kilogram, and Staphylococcus enterotoxin, 1350 nanograms per kilogram. Which one of these is more lethal? If you said botulism, you're absolutely correct. We only need 0.03 to kill 50% of the test population. Adherence is the first step in pathogenicity. The microbes must attach to a surface. Adhesions or ligands of microbes attach to a receptor in the host. So adhesion and ligands bind to receptors on host cells. Adhesions or ligands are mostly glycoproteins or lipoproteins in the cell wall, or sugars associated with the glycocalyx or fimbrine, and this can vary from microbe to microbe. Receptors on the host is usually sugars, and it varies from host to host. Example, Streptococcus mutans, which causes tooth decay. It converts the glucose to dextrin, and dextrin is used in the glycocalyx. Actinomyces attaches to the glycocalyx of the S mutants by its fimbrine. We can also see that they form biofilms. So, example of a biofilm would be dental plaque. And biofilm provides attachment and resistance to antimicrobial agents. A tapered end is used as a hook to attach to host cells. Example, Treponema pallidum, which causes syphilis. So here's a picture showing you the bacteria, and here are the adhesins or the ligands, and these attach to the receptors that we would find on the host. And this is an awesome picture showing you E. coli in the urinary bladder. So this person has a UTI. Invasion, penetration of host defenses. The capsules, which we know we find in the glycocalyx. Even though a microbe has a capsule doesn't mean it's a pathogen, and not all pathogens have a capsule. There's adherence and protection from phagocytosis if you have a capsule. So it's an immune system defense that we have to eat up anything that's foreign, phagocytosis. But these guys could be protected from that. It's often a key role in virulence, so they prevent phagocytosis. Example, Streptococcus pneumoniae is only virulent when capsule is present. And when did we see this? If you said the Frederick Griffith experiment, you are correct. The cell wall. Some bacteria have cell walls that contribute to virulence. The M protein is used for attachment and resistance to phagocytosis by white blood cells. So the M protein, resists phagocytosis. Now, M protein is also heat and acid resistant, and we find this in Streptococcus pyogenes. It also has mycolic acid, which is a waxy lipid. Mycolic acid resists digestion by phagocytes. So an example there would be Mycobacterium tuberculosis. The OPA protein that you will find in the outer membrane inhibits the T helper cells, and we find this in Neisseria gonorrhea. Enzymes. Coagulases. Coagulases causes blood clots. So it coagulates fibrinogen. Clots help to protect the microbe from the host defenses by surrounding it. So an example here would be boils that are caused by staphylococci. Kinases dissolve fibrin clots which the host may form to isolate the pathogens, so it helps them escape from the host defense. So kinases digest fibrin clots. Example, Staphylococcus uses staphylokinase. Hyaluronase dissolves hyaluronic acid, which is used to keep host cells together. So when it dissolves it, or when it hydrolyzes the hyaluronic acid, that leads to a blackening and a really horrible smell. And example of this would be gangrene caused by Clostridium. And there's another example of a kinase that would be streptokinase, and you actually see here that it produces this activated a plasminogen which causes plasmin to break down fibrin and hence dissolves the blood clot. Continuing with enzymes, coagulinase, it breaks down collagen, which you know is connective tissue, so it hydrolyzes collagen, and this helps in spreading. IgA proteases. IgA is an antibody that the host produces as a defense against invaders. Some organisms produces IgA proteases, which destroy these antibodies. So IgA proteases destroy IgA antibodies. Example, Neisseria meningitis, which causes meningitis. Antigenic variation. The antigens on the surface of a pathogen are recognized by host antibodies and defense cells. Some pathogens can change the proteins, their antigens on their cell surface, to trick the host, so that antigenic variation is when they alter their surface proteins. So an example of a virus that does that is influenza. Cytoskeleton. Actin is a protein that is a major component of the cytoskeleton. It gives the cell shape. Invasins are enzymes that rearrange the actin filaments. So for example, Salmonella alters the host actin to enter a host cell. So that's what you see in this picture. So they're altering the actin to allow them to move in. Listeria uses the actin to move from one cell to the next. Here ends Part A of Chapter 8.